disrupting nerve function in insects, leading to paralysis and death, which make it useful as an insecticide. If you see the target says toxin, toxin specifically targets nicotinamide acetylcholine receptors. We already studied in our previous classes about the nicotinamide acetylcholine receptors, how the activity will be in the insects, how the acetylcholine ligand binds to the acetylcholine receptors to enhance or to stimulate the downstream signaling pathways in order to decrease the metabolic activity of the insect or to kill the insect. And coming to the location, the receptors are found in the nervous systems of insects and other organisms or the targeted organisms. If you see the mechanism of action, toxin binds to the nicotinamide acetylcholine receptors, which disrupts the normal function of the receptors. This interference blocks the transmission of nerve impulses, leading to paralysis and eventually death of the insect. If you see the importance of toxin in pest control, by targeting NACHRs, that is nicotine acetylcholine receptors, toxin acts as an effective insecticide, protecting crops and reducing pest-related damage and disease transmission. That is the reason people are using toxin as a pest control agent or insecticide. If you see the binding mechanism of toxin, it binds to nicotinamide acetylcholine receptors by interacting with the receptors binding sites. But unlike the natural neurotransmitter acetylcholine, it prevents the receptor from functioning normally. If you see the toxin binding to nicotinamide acetylcholine receptors, it blocks the normal signaling transmission. If you see the acetylcholine binding to nicotinamide acetylcholine receptors, it allows the normal signal transmission. So, when it is acetylcholine, it allows the normal signal transmission. But in the presence of toxin insecticide, you are going to see the blocking of the normal signal transmission. That means general functions will not occur. Coming to the toxin binding mechanism, it binds to the NACHRs, that is nicotinamide acetylcholine receptors, and blocks the ion channel, as we have studied just now. Prevents the flow of ions across the membrane. This prevention or the blockage leads to the failure of nerve impulse transmission, resulting in paralysis and eventually death of the organism or the insect. Coming to the acetylcholine binding, the acetylcholine binds to nicotine acetylcholine receptors, causes a conformational change that opens the ion channel, ions to flow through. This ion flow through leads to depolarization of the neurons and the propagation of nerve impulse, which facilitates normal signal transmission. When there is a normal signal transmission, you are not going to see any problem with the nerve function. Whatever we discussed, that's what the thing was explained in the form of a flow chart. I mean, first the receptor will be activated with the acetylcholine. When the receptor gets activated, automatically the ion channels will be opened. When it will be opened, you are going to see the normal signal transmission. In the presence of toxin, you are going to see the blockage of the receptor. When there is a blockage of the receptor, the signal will not be passed to the channels. So the channels remain in a closed condition. When the channels are closed, there will be no flow of ions. Ultimately, the nerve impulse will be blocked. There is no signal transmission. Coming to the effects of toxin, neurotoxic effects of toxin, we already know that this toxin blocks the receptor. That's how it will not allow the nerve impulse. Okay, it will not give the nerve signal. Coming to the mechanism of action, the first one is toxin enters into the insect body through direct contact or by ingestion. This is the first step. And the second step is it binds to nicotinamide acetylcholine receptors at the neuromuscular junction. Okay, and the third step is it prevents acetylcholine from binding and transmitting nerve signals, which leads to continuous muscle contraction, then paralysis, and eventually it leads to the death of the insect. Okay, this is what I have given in the form of direct points. Okay, this is all about the Neris toxin. Neris toxin is a naturally occurring poison which is used as an insecticide in order to kill the pests. Okay, by regulating the nerve impulse. This is about the Neris toxin. Do you have any doubt? Hello, am I audible? Am I clear to you?
Yes, ma'am. Audible.
Tell this. Hello.
Yes, ma'am.
Okay. Now, the second one which we are going to study here is ivermectin. What is this ivermectin? We are going to concentrate about the types of ivermectin too. But here, before we enter into that, we'll talk about the ivermectin. What is an ivermectin? It refers to a group of natural compounds which are derived from soil bacterium, Streptomyces everemetilis. These compounds have revolutionized pest control due to their potent insecticidal and anti-helminth or anti-parasitic properties. Ivermectins are widely used in agriculture and veterinary medicine to combat pests and parasites, contributing significantly to crop production and animal health. Under this thing, we are going to study about two things. Under ivermectin, we are going to talk about two things. The first one is ivermectin. The second one is abamectin. The first one is ivermectin and the second one is abamectin. Coming to the ivermectin, which functionally as an anti-parasitic agent by disrupting the neural and muscular functions of parasites through its action on specific ion channels. Whatever we discussed till today, all the agents, all the molecules are acting on the nervous system of the insects in order to kill it, in order to control it. Here also, this ivermectin acting as an anti-parasitic agent by disrupting the neural and muscular function of parasites through its action on specific ion channels. If you see the chemical structure of ivermectin, it contains cyclic lactone ring with attached sugar moieties. This is the structure of ivermectin. Okay. This is the structure. I have given you different types here. This is the B1A type, B1B type. Okay. There is a slight modification in the structural formula. Here, ivermectin, whatever we discussed, the formula is it contains 48 carbon atoms and 74 hydrogen atoms and 14 oxygen atoms. But whereas here in B1B, you are going to see 47 carbon, 72 hydrogen, and 14 oxygen. There is a slight variation in the number of carbon and hydrogen atoms. This is the structure of ivermectin, and this is the molecular formula. It contains macrocyclic lactone rings with attached sugar moieties. If you see the molecular weight, this it is 875.1 gram per mole molecular weight. And then coming to the mode of action, here we already know that this ivermectin controls or regulates the ion channels by acting on the nervous system. Coming to the binding of the ion channels, ivermectin binds selectively to glutamate-gated chloride channels and to a lesser extent to GABA-rated chloride channels in invertebrates. Coming to the next point, it increases in chloride ion permeability. That is, ivermectin binding leads to an increase in chloride ion permeability across cell membranes. And this ivermectin also increases chloride ion reflex, which causes hyperpolarization of nerve and muscle cells, disrupting normal neurotransmission and muscle function in parasites. Coming to the last one under the mechanism, paralysis and death. This ivermectin lead to paralysis and death of the parasites also. And this ivermectin selectivity for invertebrate glutamate dated chloride channels ensures its effectiveness against parasites while having minimal impact on mammalian cells due to the absence of these channels in mammals. Coming to the clinical signs, in humans, it shows common side effects like gastrointestinal problems, neurological problems, dermatological problems, and systemic problems. And these are the examples which I have given under each one. Under gastrointestinal, people may suffer with nausea, vomiting, diarrhea, etc. Under neurological, people may suffer with dizziness, headache, etc. Dermatological, rashes on the skin or itching, systemic, fatigue, and weakness. Coming to the severe or rare side effects because of this ivermectin, the neurotoxicity symptoms such as tremors, ataxia, confusion, and potentially seizures in severe cases. Sometimes it may also cause cardiovascular problems where hypotension will be seen in those persons, especially with rapid intravenous administration or in susceptible individuals. And coming to the other part, allergic reactions, anaphylaxis is rare but possible, characterized by difficulty breathing, swelling of the face, throat, and hives. Now, clinical signals in veterinary. If you take dogs and cats, you're going to see the problems at neurological level, gastrointestinal level, respiratory problems, cardiovascular problems, and other signs. If you see the livestock and other animals, you're going to see similar types of effects, neurological signs, gastrointestinal disturbances, respiratory distress, cardiac effects. In severe cases, you're going to see sometimes death of the organism also. This is about ivermectin. Coming to the next part of ivermectin, I already told you, we are going to study about two different types under this. Abamectin. What is abamectin? Here it is about spectral insecticide and acaricide derived from the ivermectin family of compounds. These two, ivermectin and abamectin, are from ivermectin. Okay, we are studying these two under ivermectin. It is widely used in agriculture to control pests such as mites, leaf miners, and various insects on crops, ornamental plants, and fruit trees. Ivermectin is known for its potent activity against both chewing and sucking insects, making it a valuable tool in integrated pest management strategies. If you see the chemical structure, I already told you it belongs to ivermectin family. So it is a mixture of ivermectin B1A and B1B. When compared with, though it is a mixture of both the things, B1A is predominant component in ivermectin. Already in our previous slide, you have seen about the structures of the ivermectin. Okay. Here, these are the two structures, similar type of the structures which you are going to see in ivermectin also. Ivermectin contains only ivermectin B1A. If you see the molecular formula, sometimes it exists in the form of a mixture. Okay. When compared with the few components present in the ivermectin, B1A is predominant component seen in ivermectin. If you see the molecular formula here, it has 48 carbon atoms, which you have seen before under ivermectin, and 72 hydrogen atoms and 14 oxygen atoms. Okay, and the molecular weight is 873 grams per moles. And then toxicological effects. It shows its toxicity on humans. If you are going to see the toxicity on humans, it shows acute toxicity and chronic toxicity. Okay. Under acute toxicity, it causes irritation to the skin, eyes, and respiratory tract upon exposure. Ingestion or inhalation of large amounts can lead to nausea, vomiting, abdominal pain, and potentially it affects the central nervous system and cause dizziness and headache. Coming to the chronic toxicity, prolonged exposure to abamectin or repeated exposure to abamectin to low doses may lead to symptoms such as skin sensitization and in rare cases liver toxicity or neurotoxicity. Coming to the toxicological effects of abamectin on animals, if you could see in mammals, it shows low toxicity through dermal or oral exposure, but it can cause neurotoxic effects in high doses or sensitive species. Coming to the effect or the toxicity effect of abamectin on fish and aquatic organisms, it is highly toxic to fish and aquatic invertebrates. It can bioaccumulate in aquatic organisms, leading to potential long-term effects in aquatic ecosystems. Coming to the mode of action of abamectin, this abamectin binds to glutamate-gated chloride channels in nerve and muscle cells of insects and mites, disrupting neurotransmission and causing paralysis and eventually death of the organism. The next one is GABA-rated chloride channels. Similar to ivermectin, abamectin also interacts with GABA-rated chloride channels, further enhancing its insecticidal activity. Next, environmental fate. The environmental fate of abamectin on humans and animals, soil and groundwater, and on vegetation. We are going to study about these three things. Coming to the fate of abamectin in humans and animals. When the humans and animals are exposed to abamectin, either through ingestion or inhalation or dermal contact, primarily from residues in food, water, or direct contact with treated plants. Absorption and metabolism. In humans, abamectin is fully absorbed through the skin, but can be absorbed orally, where it is metabolized and excreted primarily in feces. And acute exposure of abamectin can lead to gastrointestinal distress in humans and animals, respiratory irritation, and neurological symptoms. And the chronic exposure may lead to sensitization and potential long-term health effects in humans and animals. Coming to the fate in soil and groundwater, this abamectin exhibits moderate to high persistence in soil, depending on factors such as soil type, temperature, and moisture content. And the degradation primarily occurs through microbial activity and photodegradation, that is through sunlight. And this abamectin has low to moderate potential for leaching into groundwater depending on soil characteristics. It can persist in soil and remain bioavailable for extended periods under certain conditions. Come to the breakdown in vegetation. The plants can absorb abamectin residues through foliage or roots, depending on application method and formulation. Once abamectin gets absorbed, this may translocate within the plant, potentially accumulating in tissues such as leaves and fruits. How the insecticides are acting in the same way, it is also acting in order to kill it. Now, this abamectin residues on vegetation can degrade over time due to exposure to sunlight, microbial activity on plant surfaces, and environmental factors. And the final part of the abamectin is environmental impact. This abamectin can impact even non-target organisms. Example, fish, invertebrates, and beneficial soil microorganisms, which does not cause any harm. Its persistence and potential for bioaccumulation in aquatic environments are of particular concern. Coming to the other part, mitigation measures, proper application practices must be followed. Adherence to recommended waiting periods before harvest and integrated pest management strategies can help mitigate environmental impacts. This is the part of abominating. In abominating, we have studied about the toxicological effects and how it acts, what is the mode of action, what parts will be targeted by this, and the environmental fail. And coming to the third one, the molecules with different chemistry, neonicotinoids. We already studied about neonicotinoids in our last class. It is slightly similar to the things what we have studied in our previous class. These are a class of synthetic insecticides which are chemically similar to nicotine. And these are derived from natural nicotine, which is found in tobacco plants. And these are widely used in agriculture, horticulture, and home gardening to control a broad spectrum of pests such as aphids, thrips, beetles, etc. And the neonicotinoids are valued for their systemic action, which allows them to apply to seeds, soil, or foliage and then transported throughout the plant, providing prolonged protection of plant from the pests. If you see the chemical structure of the neonicotinoids, which are characterized by a common structure of a heterocyclic ring, which contains nitrogen, that is similar to the nicotine which we have studied previously. These are different types of nicotinoids which we see regularly in our day-to-day life in order to control the pests. Okay. Next one is toxicity. Coming to the toxicity level of the neonicotinoids to the insects, these binds selectively to nicotinate acetylcholine receptors in the insect nervous system and causes overstimulation and disruption of nerve impulse and leading to the death of the organism by paralysis. And coming to the toxicity to vertebrates, these generally have low toxicity to vertebrates, including mammals and birds, due to the differences in the structure and function of the NACHRs, that is, nicotine acetylcholine receptors, compared to the insects. Coming to the actual toxicity, direct exposure of this neonicotinoids to higher doses can lead to symptoms such as tremors, convulsions, and respiratory distress in vertebrates. Coming to the mode of action of neonicotinoids, as we all know, it will bind to nicotine acetylcholine receptors, where ion channels are present on the nerve cell membranes. And this binding leads to continuous stimulation of the nerve cells. That's how it is disrupting the normal cell function and causes paralysis and finally the death of the insects. Coming to the systemic action, these neonicotinoids are systemic insecticides. They can be taken up by plants distributed throughout the plant via the vascular system, protecting the entire plant from the pests. Coming to the selective toxicity, if you could see the insects and the vertebrates, the selective toxicity of neonicotinoids arises from their higher affinity and specificity for insect nicotine acetylcholine receptors compared to those in vertebrates. If you see the environmental impact, sublethal doses can still affect non-target organisms like pollinators and aquatic invertebrates, contributing to concerns about their broader environmental impact. And coming to the action of neonicotinoids in insects, it disrupts the nervous system by binding to the nicotine acetylcholine receptors. They are effective against different insect pests, which causes damage to the plant. Coming to the action of neonicotinoids in vertebrates, vertebrates, including mammals and birds, have the protein acetylcholine receptors, which are totally different from the receptors which you see on insects. That's how it is showing its lower affinity for neonicotinoids. And if you could see the safety considerations, less toxic to vertebrates, chronic exposure or ingestion of large amounts of neonicotinoids may lead to adverse effects on vertebrate health. And these neonicotinoids are acting as a powerful tool in pest management due to their systemic nature and effectiveness against a wide range of pests. This is about your neonicotinoids. We will study about the toxicity and the mode of action and selective toxicity and then action in insects and action of these neonicotinoids in vertebrates. All these we have covered under this. All these insecticides are acting on the nervous system of the insects in order to protect the crop, in order to protect the plants. Okay, and against it, they are acting against the pests. And the fourth one is spinosad. Coming to the fourth one under molecules with a different chemistry, we are going to talk about the spinosad. Under this, we are going to talk about the toxicity, mode of action, and the mechanism. Actually, it is a new chemical class of insecticides which are acting against a variety of insects like fruit flies, caterpillars, mites, leaf beetle, etc. This is a non-volatile, non-systemic. It must be ingested by the insect in order to see its action. And, we all know that it is a natural insecticide and it got derived from the bacterium Staphylococcus spinosa. After the fermentation process, the bacterium to release this insecticide and that will be collected by filtration process. And this insecticide is widely used in agriculture and gardening for pest control because of its efficiency and its toxicity. Okay. Apart from that, it shows no toxicity to mammals, birds, and other most beneficial insects. And this spinosad has gained popularity as an alternative to synthetic insecticides due to its biological origin and environmentally friendly profile. If you could see the structure of spinosad, it's a mixture of spinosins A and B, which are microcyclic granules. These are the structures of spinosin A and spinosin D. If you could see the components which are present in the spinosad, we already know that it contains these two components, spinosin A and D. These are the complex molecules with multiple oxygen containing functional groups and a unique tetracyclic ring system. This spinosad contributes to its stability. The complex structure contributes to its stability and effectiveness in pest control. And if you could see the toxicity, this shows no toxicity when ingested by mammals, no additional adverse effects from chronic exposure. And it is slightly toxic to birds, moderately toxic to fishes, and slightly to moderate toxicity to aquatic invertebrates. However, it is slightly toxic to bees, oysters, and other marine mollusks. Coming to the impact on beneficial insects, It is safe for most beneficial insects. Spinosad can still affect non-target organisms, emphasizing the importance of targeted application and integrated pest management practices. Coming to the mode of action, this spinosad acts on the insect nervous system and it causes the overactivity through the receptors, cytolin receptors, and this spinosad binds to the cytolin receptors and acts as an agonist, leading to prolonged opening of ion channels and hyperexcitation of the nerve cells. And this lead to rapid nerve depolarization, paralysis, and ultimately death in susceptible insects. If you could see the clinical signs, sometimes there will be respiratory problems, there will be decreased activity, poor weight, and loss. All these things we could see in different organisms because of this spinosad. Though it is a natural insecticide, but there are some effects which we can see with respect to mammals and other organisms also. This is all about your molecules, the molecules with different chemistry. We have studied 4 different types today. Here, the first one, here we have studied this Aristotle. The second one is ivermectin. Under ivermectin, we have covered 2 things, abamectin and ivermectin. And the third one is neonicotinoids, and the fourth one is spinosad. This is all about your molecules with different chemistry. And coming to the next part, we are going to talk about these things in our next class. Hello? Sorry, hello? Hello?
Yes, ma'am.
Yes, ma'am. Am I disconnected in between?
Yes, ma'am.
At where exactly? Hello? Where did I disconnected? In which part?
Madam, not voice is not clear, ma'am. Actually,
Am I clear now?
some other sounds are coming in the middle.
Where I got disconnected, can you please let me know?
Toxicity of spinocytes.
Did I complete the toxicity of spinosad?
Yes, ma'am.
Okay, sorry. I didn't realize. I'll give my number. If there is any interruption, please let me know. Okay?
Okay, ma'am.
Please make note of my number 958. Hello? Hello?
Yes, ma'am.
Yes, madam.
958-170-4244.
Madam, please, could you please repeat it once again?
Yes, 958
958
170
170
4244.
Madam, after 170,
4244.
4244.
Yes, Amma.
yes, ma'am. Thank you.
Okay. If there is any interruption, please let me know. Okay.
Yes, ma'am.
Okay. Actually, I have a network glitch here. So I didn't realize that I got disconnected. Sorry. Now, coming to the spinosad, we already discussed these are a new chemical class of insecticides which found effective against a variety of insects, including fruit flies, caterpillars, spider mites, and leaf beetle larvae. And this spinosad is a non-volatile and non-systemic. And these are derived from the fermentation of bacterium Saccharopolyspora spinosa. It is widely used in agriculture and gardening for pest control due to its effectiveness against a broad spectrum of pests while being relatively low in toxicity to mammals, birds, and most beneficial insects. Can you see the screen?
No, ma'am. We are not able to see the screen.
Oh, ma'am.
And sorry. Right. Okay, and if you could see the structure of the spinosad, it is a mixture of spinosine A and spinosine B. These are the complex molecules which contains oxygen, multiple oxygen atoms with the functional groups and unit tetracycline system. And coming to the toxicity, it shows low toxicity towards mammals, birds, and humans. And it has a favorable safety profile compared to many synthetic insecticides. And if you could see the impact of bispinosad on the beneficial insects, it is safe for most beneficial insects, but can still affect non-target organisms. And coming to the mode of action, this spinosad acts primarily on the insect's nervous system by targeting nicotinic acetylcholine receptors. This spinosad binds to the protein acetylcholine receptors and acts as an agonist, leading to the opening of the ion channels and hyperexcitation of the nerve cells. This spinosad leads to the disruption of the nerve depolarization, disruption, and lead to the nerve depolarization, paralysis, and ultimately death of the insects. So this is all about the spinosad. So in the spinosad, we have covered the toxicity, mode of action, and the metabolism. Metabolism. And today we have covered about the Lewis toxin, ivermectin. Under ivermectin, we have studied ivermectin, abamectin, and then neonicotinoids, and then spinosad. We have covered a total of 4 topics today. In the next class, we'll try to cover the remaining 6 topics which are left under this unit, molecules with different chemistry. And if you have any doubts, please ask me. I'm sorry, I don't know.
No, ma'am.
Disconnected. Is
Hello,
it?
ma'am.
Hello? Am I audible?
Yeah, ma'am. Ma'am, a
Yes.
small request.
Yes.
Please take more class on Sunday and less class on Saturday because we are hoping, ma'am, we could not hear the class. Means we could not attend the class.
Okay, okay. I don't know about this.
Uh, yeah.
Okay.
More important topics, please cover on Sunday.
Sure, sure. Okay. Anyhow, I'll provide you the notes also. As you have informed me, I'll try to take more time on Sundays compared to Saturdays. Okay. Anyhow, I have
Okay,
given.
ma'am.
Please contact me if you have any doubts, and we'll try to connect tomorrow.
Okay, ma'am. Thank you.
Okay, ma'am. Thank you, ma'am.
Is it helpful for you? Did you have a clear picture?
Yes, ma'am.
All the insecticides are acting on the nervous system of the insect.
Nervous system of the insect.
Okay, if you know the mechanism, then that will be easy for you to understand. In our last class, we have studied the mechanism with the pictures. How exactly it does. If you could understand that, everything will be easy for you. Okay?
Okay, ma'am. Okay, ma'am.
Okay, ma'am.
Shall we leave for the day?
Yes, ma'am.
Okay, ma'am. Thank you, ma'am.
Thank
Madam,
you.
please share the PPT in the group, ma'am.
Yes, Amma. Once it is done, I said that I'm going to share the PPTs. Unit wise, I'll share the PPTs and the notes. Okay?
Okay, madam.
Okay, ma'am. Thank you.
Okay, ma'am. Thank you. Bye. Considering the phone, you said.